Okay, so my presentation goes very well after the first presentation, actually. I believe one of his exact sentences was, so today I'm going to talk to you only about neurons. There's another type of cell in the brain, but I'm not going to talk about it. But you did, actually. You did. Actually, a lot of your presentation was about it. So I'm talking today about the other brain cell. And 3 guesses as to what this structure is. Any guesses from the audience here?
I'm not going to say it.
Yes, in the back.
So, is the zebrafish?
Zebrafish? Close. In what part? Everybody
It's the
is
brain.
silent. Yeah, it's the brain of a fruit fly, actually. And I've gotten many other suggestions. I don't know where people get ideas from, but other suggestions. 3 colors here. And in the red, you can see what are the neurons. And here you can see what makes up the eye. You know how fruit flies have 100 million eyes? They have all those eyes here are the red dots. Blue is the nuclei, and in green, you have the other brain cell. So, what do you think is in your brain? We all have already heard about neurons. How many neurons do we have in our brains?
A lot.
Right. Yes, we have 100 billion neurons in our brain, generally, about. I mean, people have not counted actually every single one. And so in our brain we have what was talked about in the last talk, cerebral cortex, which is this sort of higher functioning part of the brain. And then we have the cerebellum, which is sort of the instinctive part, where your breathing and balance and general life functions are. So what do you think the divide is between the number of neurons in the cortex versus the cerebellum? Who thinks the cortex has more neurons?
It's bigger.
It's bigger, yeah. And who thinks the cerebellum has more neurons? Awesome. Well, I would say the cerebellum actually won there, and you guys are right with our 20% voting ratio here. So yeah, there's a lot more neurons in the cerebellum, a lot more neurons in the cerebellum, even though the higher functions are in the cortex here. However, for the other type of cell in the brain, how many types of, how many cells do you think we have of this in our brain?
500.
More than a lot.
More than a We actually have about the same amount as neurons. And the interesting thing is that we have way more of these other types of cells in this cortex here versus in the cerebellum. And so the other type of cell is called a glial cell. And glial cells have many functions, one of which was described in the last talk, actually, where there was the ventricle here, where there was the cells that migrated up. And what he didn't say was that these radial cells that are the ones that give rise to the neurons that then migrate to the other places in our brains, these cells are actually glial cells. And I'm not sure if somebody can press play on the screen. There's a nice little cell here. This is a neuron that's migrating along.
It's a touch screen, so you can just literally go on the screen.
Yeah, touch,
Oh, there,
Don't be
okay.
very light.
play.
Ah, yeah, it is.
There you go.
There.
You just need to...
It's alive!
I don't know how many people saw that, but this neuron migrated from here to here. There you go. And so this neuron is actually migrating along glial cells. So one of the functions of glial cells is to direct neurons where to go. Another function is to act as sort of the third wheel in the neuronal synapse. So everybody knows, or often people know about how the neurons contact each other and they send neurotransmitters from one neuron to the other neuron. And this is sort of how you send signals around in your brain and also to muscles. And recently it's been shown that the glial cells are sort of right beside there and regulating this transmission between these two, the messages between the two neurons. And so here we have an image of a green glial cell with a red neuron, and you can see all of the different arms of the glial cells that are able to touch many different neurons. So another function of glial cells is to sort of regulate the function of neurons. And this is where you can imagine where in the cortex you have tons of glial cells regulating all of the higher order functions of human brain. And the last function that I will talk about today is glial cells insulate neurons. And this is something that people may have heard about myelination, which is important in diseases like multiple sclerosis, where if the glial cells are affected or the body attacks the glial cells, then the insulation around the neurons gets disintegrated. And so what a signal which you have from like your brain to your muscle actually doesn't get there because it loses its signal along the way. And so when I was an undergraduate student, I heard about these cells that wrapped around an axon, so this long part of the neuron here. It's called the axon, and they wrap around it and make all these layers. And I thought, as a undergraduate student, that is so cool. I want to study this octopus-like cell and study how it wraps around. And so then I spent the next 6 years of my life looking at this. And so this is what it looks like when you zoom up using an electron microscopy. But here basically you have a whole bunch of neuron bundles, axons going down to the muscles. And so I actually used fruit flies. That image from the beginning was of a fruit fly brain. And the reasons that we use fruit flies are because instead of 100 billion neurons, there's only 100,000 neurons. Which actually, can somebody press the screen again? This is just this year, they have mapped where every single neuron in the fruit fly brain comes from and where it goes. And so this is really useful when you're trying to figure out which neurons interact with each other. And the real reason I got involved with fruit flies was because there's crazy, awesome, fun, fluorescent experiments. And so I really just tried to put as many fluorescent images in my presentation here as possible. And this is basically why I got a PhD in the first place, was because I wanted to be on the microscope. And success! Did it. And so this is the peripheral nerve of the fruit fly here, and these black dots are all of those axons of the neurons going down in a bundle to the muscles. And then you can see that there's 3 layers of glial cells around. There's the blue layer, the orange layer, and the green layer. And then outside of all of this is something called the ECM, which is also known as connective tissue, where this is all of the different proteins that make sort of this matrix. And this is what it looks like. It's in a cartoon super zoomed up. You have all of these different proteins in a big matrix outside, and they actually bind to the cells that they are on top of, where there's receptors. And so you have these connective tissue binding to receptors on the glial cells. And this causes a lot of downstream or internal signaling in the neuron and the glial cell. And specifically to the actin filament, which is like it's called like the skeleton of the cell. And so what you have is this connective tissue binding through receptors in the membrane of the glial cell to the actin cytoskeleton. And so the research questions that I had were, why are the laminin, which is one of these specific connective tissue proteins, why are mutations in laminin worse in humans than mutations in the receptor? If something binds one thing to another, you would expect them to have about the same effect. And the other one is this protein called basigin, a receptor, does it actually bind to this protein laminin, this specific protein? So basically I'm looking at this interaction here of the connective tissue going through the receptors to the glial cell. And so if you take this cartoon and look at it in a real fruit fly, here you will have all of this is a control organism, and you have all of the axons here with the purple wrapping around. So here it's like a cross section of a nerve. So you have the glial cells wrapping around all of these blue axons with the other layers here. And when you can see that there's no receptor, then the glial cell doesn't wrap around anymore. The purple cell here, it actually, and this means that all of these axons the neurons are right beside each other, so when one fires, it's gonna send its electrical energy signal into the other one, and so you're gonna mix up your firing. You're gonna try to move your left leg and actually move your right arm. And so that is not very good for the fruit fly. Luckily, we're only dealing with fruit flies, don't worry. And so the other thing that I looked at was this really awesome thing called lifefact, which is basically the actin cytoskeleton in fluorescent green. And so in the controls, you can see that the actin cytoskeleton goes around the pink neuron here, the nerve. It's very like uniformly distributed. It goes nicely around the whole thing. This is just a black and white image of the green section here. However, when you don't have the receptor anymore, then you get this, it's only on one side here, and it's actually all clumped up. You don't have this nice looking actin cytoskeleton. And so basically it results in the condensation and the collapse of the actin cytoskeleton. And then what does that actually mean for the fruit fly? Well, if you have this nice brain structure here with these nice nerves of the control fruit fly, when you don't have a receptor, the brain is all condensed. And so it's basically like an elastic band that you've taken and stretched, and then it like retracts. And so you can actually, this is what I thought was really cool. These are the moments I lived for, where you have this like, the sort of protrusions from the membrane going out, and it is just like, no cell should look like that. It's really messed up, which is what you're always looking for. So that's what I was always looking for. You can just, fruit flies are the best because you can do all sorts of fun, crazy experiments and just try to get the prettiest, craziest mutation you can. So then I wanted to see, did the same thing happen if I mutated the connective tissue? So now I've mutated the receptor, does the same thing happen if I mutate the connective tissue? And so this is what it looks like with, this is another control. You can see beautiful wrapping of the glial cells around the axons here. And this is what happens when you don't have laminin, a specific form of laminin. This is not actually this, well, okay, so there's a size difference here. You can see how big an axon is here. It's the same size here. It's just the cell, the glial cell, is so swollen. And so this is what I found here. And it's important to know that there's, laminin is made up of 3 proteins that then like sort of, they all intertwine together to make one functional protein. And so just keep that in mind as I'm talking here. But I mutated one of them, one of the 3 subunits. And so I wanted to figure out what was in this like really swollen area of the nerve of the fruit fly. And so here you can see this is a control again, and you have the axon surrounded by this is an electron microscopy image, so you have the black dots are the ribosomes, which are what is important for like making new components inside the cell. And what you can see, even if you don't really know what the black dots are, you can see that there's a lot more here, and this whole area here is just all ribosomes. And so there's really something going on in this cell that is involved, that is really affecting the ability of the cell to make new proteins. And so what I found was that the electron microscopy revealed that there was increased ribosomal content and swollen endoplasmic reticulum, which is a word you might remember from like grade 12 a long time ago, which is where new proteins are made. And so in the endoplasmic reticulum, the way that a normal sized protein, because connective tissues are really big, they're huge, fibronectin, they're fibrous, they're really big. Most proteins are very small relative to these, and they get secreted nicely in these very uniformly shaped vesicles from the endoplasmic reticulum to the rest of the cell. And with these long extracellular matrix or connective tissue components, they actually have to bind to this other protein that then tells the sort of spherical vessel, like, hey, I'm really big, you have to slow down, you have to take more time to get out of the endoplasmic reticulum. And so then you get this nice big vesicle with these big connective tissues, so that goes to the outside of the cell. And so when you eliminate that special factor that tells it to like slow down and make this big vesicle, you find that you do actually get these areas. So this is a peripheral nerve of the fruit fly, and in the purple here you have laminin, and you can see that it's actually stuck in these clumps inside the peripheral nerves. It's not nice and uniform along the outside. And what you can also see is in this one, this is science for you. The little dots here, where the purple and the green overlap, that is bang on. Which you can't see because of the resolution, but basically it's showing that the laminin is indeed accumulating inside the endoplasmic reticulum, and this is important because of the way that the laminin gets secreted. So now we're trying to figure out why is the laminin stuck inside the endoplasmic reticulum when you mutate one of these subunits here. So when you mutate one, why does the other one get stuck inside? And And so so I'm working in fruit flies, but somebody before had studied this in tissue culture, so in cells that are not really anything in particular. And sorry. And basically they found that when all three of the subunits were made properly by the cell, then they joined up, they became really nice all together, and they were secreted properly. But when only one was made, then the other ones, this alpha one would get secreted by itself, but the other one would get stuck inside. And so basically there's always one stuck inside if you don't make all of them. And so what I wanted to look at was, okay, is this the reason that I am seeing such a big problem when I mutate one of these proteins, one of these subunits in the glial cell? Is it because the other one is getting stuck inside? Is that what's happening? And so this was an experiment that I didn't do for 2 years because my professor told me to do it, and I was like, no, I refuse. My life is worth more than measuring the width of hundreds of fruit fly nerves. But then one day I decided in about year 4, okay, maybe I should probably do this. And so I did, and sure enough, there you go, you have your paper, everything's ready because brings everything together. And so what I found out was that yes, the width of the fruit fly nerves was wider when there was no laminin, but that you could make it smaller again if you got rid of the other extra laminin that was inside. And so you're like saving the fruit fly that way. And then here I also showed that the speed, so then I did another fun experiment where I measured the speed of the larvae into the, you put them in the middle and you measure how far it takes them to go to the outside. And I did a lot of this. This was actually, I didn't have to measure it, there was a computer system that did this, so that was much better. And I found that, yep, when I mutated them, they compared to controls, there was no movement. However, I could rescue this with decreasing the other monomers. So yeah, this is a good little bit of science. And in summary though, the important lessons to take away from this are that glial cells are important. They have many functions, and that even just one receptor in glial cells, like the receptor I was talking about that resulted in the lack of wrapping and the condensed grain of the fruit fly, even just one receptor in glial cells can affect the entire architecture of the nervous system. And also that proper secretion of the connective tissue is important for the glial cells. And this is important because there are some mutations in humans where humans have a mutation in one of the connective tissue genes, and there's sort of a long-term effect on the glial cells. And this might be one of the reasons that there's this delayed glial effect. And so here is just another cool image of a fruit fly brain with the this receptor in green. And the other cool thing about doing experiments in fruit flies is that we don't, this gene is in humans, we have no idea what it does. And we don't know what it does in fruit flies. And here it's in the neurons, it's in the glial cells. We have no idea. So this is kind of the cool part about working in fruit flies. And here is the front cover of the Journal of Neuroscience where my research was published. I sent them a whole bunch of pictures that were very relevant of my research, and this one that had nothing to do, I don't even know what these little green bulbs are, and there was that one. Great. And here's laminin, good old laminin. So with that, I'd like to thank all of you guys for being here. Thank you for laughing along with me. Thank you to Cafe Hugo and of course Anna. And thank you to everybody who helped me while I was still in Canada, teaching me all sorts of cool stuff. Yeah, thank you. Yeah. Yes.
So, if I may, 2 questions.
Sure.
In one of your electron microscopy images, it seemed as if it wasn't just the glial cell but also the neurons which were affected. And could you explain that a bit more? Is it because of the glial cell that the neurons are also affected?
It would only be because of the glial cell, because the way that I did my experiments was we mutated, we didn't really mutate, it's just easier to say that. We actually knocked down a gene only in glial cells, and so the neurons are perfectly normal. And if you see an effect in the neuron, that means that it's from affecting the glial cell, which then affects the neuron, which is obviously going to happen. I mean, if they're not moving around, the neuron is not working. And so, yeah, so to answer your question, it's from the effect on the glial cells.
And the second one, more importantly, did you have these crazy contests about who could dissect more flies per hour?
No, we didn't.
No.
Damn. How could you not have done that? Yeah. We did have crazy contests in the fly room of like when you have to flip the flies to new food, and you have to sit there for like 3 or 4 hours and flip all of you had vials of fruit flies into new food, and it just becomes your habit. Yeah.
Thank you.
Yeah.
Last question.
Or more like a basic research question. So as far as I remember, the myelin or the glial cells is mostly around the axons.
Yep.
What about the dendrites? As far as I remember, not much is around those parts.
Yeah, yeah, you're right. That the main function of glial cells is to insulate the axons, because that's where the electrical charge is going, like the action potential is going down the axon, whereas the dendrites don't really function using action potentials. So there's less, but I haven't heard of any time where there's myelination on the dendrites. I don't want to say for sure that it doesn't happen, but I know that it's a lot more on the axons. Yeah. Good question.
Any more? No? Okay, so let's thank Lindsay again. Thank you very much.